This research is on investigating the cytotoxic effects of flavopiridol on MCF7 breast cancer cells. Breast cancer is the most common cancer in females in the United States and is associated with the second highest death count due to cancer in females worldwide. Breast cancer has three receptors, one for the female hormone estrogen, one for the hormone progesterone, and one for the human epidermal growth factor protein. Therapeutic drugs are often developed to target these receptors. Flavopiridol is a crystalline semi-synthetic flavonoid that is obtained from 4 different indigenous plants in India. It is a major inhibitor of cyclin-dependent kinases, which are involved in regulation of the cell cycle and are considered as important protein targets for designing anti-cancer drugs. Flavopiridol exhibits antitumor activity in the form of CDK inhibition, apoptosis, DNA interaction, and cyclin D1 decrease. It is known to inhibit CDK7, 8, and 9 inhibitors. These are kinases responsible for transcription, and as a result, this inhibition can induce apoptosis and inhibit proliferation and/or viability of estrogen receptor-positive breast cancer cells. Now, ER-positive breast cancer cells, like MCF7, have anti-apoptotic proteins. In this research, I explored the expression of MCL1 as it is the most frequently amplified anti-apoptotic member of the BCL2 family. Flavopiridol induces apoptosis in cancer cells through the activation of E2F1 transcription factor and repression of anti-apoptotic MCL1. So, other than inhibiting anti-apoptotic genes, flavopiridol also induces p53, which is an apoptotic gene. It does so by inhibiting the transcription of MDM2, a short-lived mRNA, to prevent the degradation of p53, thereby inducing p53. In this research, I aim to determine the effects of flavopiridol on cytotoxicity in MCF7 cells and to assess any effects of flavopiridol on the expression of p53 and MCL1. I did this by performing a cytotoxicity assay, the MTT assay on MCF7 cells after they were treated for 24 hours. Different doses of flavopiridol were tested to determine a dose that was cytotoxic significantly. At first, the vehicle control was responsible for the decrease in cell viability, as we can see here. So next, percentage of DMSO was reduced for vehicle control and PBS was used for diluting the samples. Surprisingly, it led to an increase in cell viability. This could possibly have happened because the wells containing the cells were highly confluent and out of the detection range of the MTT assay. Next, I tried higher doses of flavopiridol and successfully saw a decrease in cell viability without the vehicle control affecting it. So I chose to analyze the effect of the 25 and 100 micromolar doses on MCL1 and p53 gene expression. qPCR analysis revealed that flavopiridol treatment led to a downregulation of anti-apoptotic MCL1 gene. However, it was not significant. Now looking at the p53 expression, we saw that the 100 micromolar flavopiridol treatment led to a significant upregulation in apoptotic p53 expression, suggesting that cell cytotoxicity may be via the p53 pathway. Next, we wondered if flavopiridol is cytotoxic to healthy cells, that is, the healthy CHOK1 cells. Unfortunately, all the doses ranging from 1 to 100 micromolar were cytotoxic. In conclusion, we successfully demonstrated the upregulation of apoptotic p53 in MCF7 in response to flavopiridol treatment. In the future, the effect of flavopiridol should be observed in another anti-apoptotic gene in the BCL2 family to see if it gives different results than what we saw here in MCL1. Furthermore, a flavopiridol dose that is not toxic to healthy CHOK1 cells has to be determined in vitro, followed by in vivo testing and human clinical trials. A dose between 0.1 to 1 micromolar seems promising according to our data and should be tested on both MCF7 and CHOK1 cells with longer incubation times to figure out a dose that is selectively cytotoxic to MCF7 cells only.